नीचे अपना मोबाइल शांत कर लेना.
A very good morning to all of you. Welcome you all in ISFCP Dialogue Series under the aegis of IQAC-IIC ISFCP, organized by ISF College of Pharmacy and Autonomous College Moga, Punjab. Today's guest and speaker, Professor Dr. Nitin Sharma, HOD Department of Pharmaceutics, and his title of talk is Radiopharmaceuticals in Product Development. Sir, we welcome you in this dialogue series and requested you to please proceed further.
Thank you, Dr. Mehan. So, in today's presentation, we are going to learn a very new technology, very innovative technology for the product development. As you know that nowadays the pharmaceutical company are keenly working on the product development. They want whatever the in vitro data they are generating in their laboratory, these data should mimic completely with the in vivo efficacy or in vivo performance of that pharmaceutical product. But anyhow, this is quite difficult to correlate the generated in vitro data with the in vivo performance of these products, which are affected by the several factors. I'm not going deeply on these factors, but how nowadays the performance of any pharmaceutical product can be determined or can be monitored with the help of the radiopharmaceuticals that we are going to learn today's lecture. So thank you once again, Dr. Mohan, for this opportunity. With your permission, I am going to define all the procedure, all the aspect, all the opportunity in this lecture. So the concept is, this is not a novel concept. However, the application of this concept is the novel. Radiopharmaceutical, we are already being used for the diagnostic purpose, as we are going for the X-ray whenever we are meeting with the accident. That is the application of the radiopharmaceutical, but not for the product development. This application is for the diagnosis purpose, at which area or which part of my body is damaged or the bone is damaged. Similarly, if I am going to the PET scanning, CT scanning, that all are involving the application of the radiopharmaceuticals, but they are only for the diagnosis purpose. Rarely they are using the pharmaceutical product development, but nowadays we are fortunate enough that now we are having the techniques which can be used radiopharmaceuticals in the product development as well. We can determine how my product is, how the performance or how the product is performing inside the body after the consumption of any pharmaceutical product. So this is the whole concept. There is the science behind the ligation of the radiopharmaceuticals with the targeting moiety. We know the generation of the disease is involved some overexpression of the certain kind of the receptor into the body. Like if I'm talking about the cancer, so this cancer is, whenever the cell is generating the cancer, so there will be the overexpression of the folate receptor or the various kind of the receptor on the this cell. So this is making distinguish the cancer cell with the normal cell. So if I am able to target that specific receptor on that specific cell which are generated by the response of the generation of the disease, it's mean we can specifically target that specific cell or further the tissue or the further the organ, right? This is the well-known concept. But what we are doing, we are ligating the radiopharmaceuticals with that targeting molecules with the help of the linker. So there is some chemistry behind. Normally we are using the technetium as a radioactive material, the stannous chloride as a reducing agent, or it is act as a linker, and the targeting moiety, it could be any protein, or it could be any pharmaceutical product, it could be any formulation. Okay, whatever my objective, like if I am going to determine how my product, like how the drug is going to bind with that particular receptor, so in that case the targeting molecule with my therapeutic product. Similarly, in case of the monitoring the performance of so-called targeting device, like we are preparing the nanoparticle, we are preparing the liposomes, erythrocytes, there are multiple targeting moiety or the formulation we are generating in the laboratory. I want to determine at which rate that prepared formulation is ligating to that particular or targeting to that particular cell. So in that case, this targeting molecule will be that specific formulation. It could be the nanoparticle, it could be the liposome, and anything. So my therapeutic substance directly it could be the targeting molecule. My formulation that it could be the targeting molecule, or any kind of the protein that could be the targeting molecule. So with the help of a chemistry or this linking will be done, the ligation will be done. So this complex, what happen when we will consume this complex? So here this targeting molecule will navigate this complex which are having that radiopharmaceuticals, and by the response of the navigation, so this targeting molecule will attach to that specific receptor which are overexpressed on that particular cell in case of the particular disease. So this binding will tell ki what is means what is the performance of my product, right? Because molecule is binding to that particular receptor, and this molecule is binding with the that particular radioactive substance, and radioactive substance is continuously generating some rays, and an external camera is determining the from which area these rays are coming from, and from where we are getting that rays. It's mean that targeting moiety is attaching to that particular area, which are indication that specifically that area is my region of interest where this targeting molecule is attached, which is concluding that either specifically the cancer location, or it could be the conclusion of my prepared formulation is directly reaching to that particular site. So with the help of this technology we can determine several kind of the targeting efficiency, including the performance of even the biological product, that we can determine at which rate the biological product is going inside the body, or where it is going, at which rate it is traveling. So there are the numerous advantages of this technology. Like we are working on the nose to brain delivery, so we can determine whatever the compound we are administering into the nasal cavity, whether it is reaching to the brain or not. We are working on the cancer molecule targeting, so with the help of this technology we can determine actually that so called targeting molecule is reaching to the particular site or not. So this is the beauty of this technology. So more specifically, give me 1 minute because my slide is not moving. Yes. So more specifically, this is the concept. This is my drug molecule. I am ligating this drug molecule with the help of this radio pharmaceutical product or the radioactive substance. It could be anything. It could be the technetium, which are normally using in this case. It could be the cobalt. It could be the indium. It could be the iodine. Any kind of the radioactive material we can use, depend on its biological half life. So after the radio labeling, this whole complex will call the radio labeled drug. So this is the prepared formulation. Like in case of here, I am preparing the nanoparticle, or additionally it could be the liposome, it could be the any conventional formulation, even the tablet it could be, or the aerosols, or the diprotoannular, any kind of the formulation this could be. So now in the next step, I will incorporate that radio labeled drug in that particular formulation. So this will call the radio labeled drug embedded in the drug delivery system. And after is its administration into the body, it could be the animal or it could be the human body. So an external camera will capture the image, and we are going to get these kind of image. Like in this case, this region of interest is this highlighted brown color area is the region of interest. So this is including or concluding that this is the application site. In this case, we administer the IV through the IV route we administer. So after the application, my drug or my formulation is circulating at this area. Here the liver, this is the gallbladder, the some portion is accumulated into the GIT. This is the urinary bladder, means after the means drug is continuously eliminating, and it is accumulating at the urinary bladder, and it is ready to eliminate from the body. So in this case, we can determine at which rate my drug is circulating or the biodistributing, and at which rate the drug or the formulation is eliminating from the body. So the PKPD study we can determine with the help of this gamma scintigraphy. And this technique is providing some visual images. So these are the real-time performance data we are getting. So this is the beauty of this technology. All right. Now I'm going to the case study where this technology can be implemented. So in case of the oral product, like if we want to determine its disintegration time or the site, suppose I am preparing the colon specific tablet. So on that case, my objective is my formulation should not be released at the upper GIT, and as soon as it is reaching to the colon site, all the pharmaceutical product or the therapeutic substance should immediately release out. Okay, on that case, this technology can be very useful. Similarly, the gastric empty rate, or at which rate my product is traveling inside the GIT, the drug release behavior we can determine. Suppose if I am preparing the GRDDS gastroenteritis drug delivery system, on that case there is approach mucoadhesion. So whether my prepared formulation is adhering at particular site or particular location, that we can determine, or how long my formulation is adhering with the biological membrane, that we can determine. What are the effect of the food? Suppose if I'm consuming my formulation along with the fatty food, and the same formulation if I'm consuming in the fasting conditions, so what will be the effect of this food content on the performance of this product, that we can determine. As I already told you, the PKPD study we can determine, and the disintegration behavior or the bioavailability correlation, in vitro, in vivo correlation, that also we can determine with the help of this gamma scintigraphy technique. So here we are representing one of the performance of one of our product. This was the colon specific tablet, and these are the image of the different time interval. This is the 0 minute image. Here it is clearly indicating that the prepared product is still in the stomach. As we are moving or the time is moving, the product location is continuously changing, but still at 240 minutes it is reflecting that product is still in the small intestine. But as soon as I am just evaluating the image of the 480 minute, it is showing that the formulation is at the ileocecal junction. This is the initial part of the large intestine, but still the drug is embedded inside the tablet. This is not started to release the drug. Okay, but at the 440 minute, the drug release now started, and at the 600 minute, the whole colon is filled with the drug, which are the clear indication that whatever the product we have prepared, that is preventing the drug release at the upper GIT, but allowing the drug release at only specifically to the colon side. So whatever the colon indication, like these kind of the formulation can be very useful in case of the ulcerative colitis, in case of the colon cancer or the inflammatory bowel disease syndrome, any kind of the disease which are specifically related to the colon, where this formulation can be used. And with the help of this gamma scintigraphy technique, I can be visually very sure that my product performance is very at high level. So as a product development, if I am having these kind of the images which are showing the product performance into the body, so that is very good impact in front of the regulatory. Means I can be very much sure, or I am having the power to convince the regulatory body that my product is specifically for this purpose, so please provide the approval of my product. So this is in terms of the regulatory, this technique is very useful. Similarly, in case of the pulmonary product development, there is continuously the mucociliary clearance, so whatever the pulmonary product we are using, like in case of the tripod inhaler, in case of the aerosol delivery, in case of the any kind of the pulmonary drug, whatever the product we are consuming, there is a mucociliary clearance immediately, right? Although this is the defensive mechanism, but anyhow, these product are also captured by the mucociliary clearance. For minimization of this mucociliary clearance, we are using the number of the strategy, like we are increasing the viscosity of the formulation, We may use the mucoadhesive polymer into the formulation. So after this approach, whether the approach is suitable for making a effective product, that we can determine with the help of this gamma scintigraphy techniques. So the lung deposition pattern we can determine with the help of this where actually my formulation is uh, depositing, whether it is upper lungs or the lower lobes, whatever. What is the residential time of my formulation, right? As I told you, there is a mucociliary clearance. So if I'm using any specific kind of the approaches, so at which level this approach is working, how long my formulation is remaining to the lungs, because overall retention time or residential time of any formulation is regulating its bioavailability. So it is very, very important that my formulation should be retained for longer period of time into the lungs for better performance. So how long this formulation is retaining inside, that we can determine with the help of this technology. So overall, finally, with the application, the device performance is spacer performance, we can determine. We are having the multiple kind of the device for the application of this pulmonary product. So how perfectly my device is able to deposit the formulation inside the pulmonary route, that we can determine. So here are some images. In case of the salbutamol, we prepare the microsalbutamol and the nanosalbutamol, and the image are clearly indicating that in case of the microsalbutamol, the major portion means 25% of the drug was retained on the esophagus region only, and there was a minimum part of the drug retained in the lungs. But in case of when I prepared the nanosalbutamol, the minimum part was retained in the upper pulmonary route or the esophagus, and the majority portion, 80%, 81% majority part was deposit on the lungs. So this is the beauty of this gamma scintigraphy. However, there is no technique available which tell about how much portion of my prepared formulation will adhere at which location on the pulmonary rate. So this is a very good indication. I can determine, or we can very much sure get where my drug is reaching, or what the portion of my drug is uh, locating at my desired site. Similarly, this is the pulmonary delivery or device performance. The same product was used, but delivery was done with the three different device. In this case, this was the device one, and all these three images, it is clear indication that product deposition pattern are different, entirely different. In case of the device one, this is only delivering at the esophagus region. In case of the device two, some portion of the drug is reaching to the lungs, but still some drug is at the esophagus. But the device three performance is extraordinary. Majority portion is reaching to the my desired site. So this is again the beauty of this application. As I told you, the mucociliary clearance is again the defensive mechanism, also, or this is not something which I like in case of the product, the pulmonary route. So if I want how much my strategy is useful to retain or increase the residential time into the lungs, that we can determine. We are capturing the different time. We are capturing the gamma images at different time. Like this is the 0 minute. So here the intensity of the region of interest will tell the percentage of the drug deposition, and you are continuously, at by increasing the time, the intensity of the drug are continuously decreasing. That is due to the performance of mucociliary clearance. Means after the deposition, immediately 360 minutes, your almost the majority of the portion of your drug is either absorbing or eliminating by the response of the mucociliary clearance. So rate of mucociliary clearance we can determine with the help of this pulmonary route. So these images are the clear indication the product performance. Similarly, in case of the topical product, we can use the gamma scintigraphy for the skin penetration, for the skin permeation. Like in the case of the topical product, we want the mystratum corneum should be bypassed. So whatever the strategy I am using, whether I am preparing the liposome or I am preparing any kind of the approach, whatever the approach we are using, at which rate this approach is successfully delivering the drug inside the skin, that we can determine with the help of this gamma scintigraphy technique. Skin deposition and the localization portion means specifically at which area, whether this is epidermis or the dermis on the endodermis, where my drug or the formulation is residing, that we can determine with the help of this gamma scintigraphy technique. And this is very, very useful in the case of the transdermal, in case of any kind of the topical drug product. So this is one of the study which we perform for the gel. This was the Mucociliary gel. These spots are indicating the product application, and as the time is increasing, here are the decrease in the intensity of all the spots which are indicating that product is transporting from the stotan corneum to the dermis and to the lower part of the skin. So this is very good visual inspection or monitoring of the product performance. Similarly, in case of the parrot, if you are preparing any parental product and you want to determine its pharmacokinetic, that we can determine the clearance from the intramuscular intraperitoneal route, that we can determine pharmacokinetic Cmax, Tmax, that we can determine with the help of this gamma scintigraphy technique. So overall, any kind of the product performance we can determine, like if you are preparing the ophthalmic products, and that is specifically the nasolacrimal drainage is again the clearance mechanism. So whatever the strategy you are including to eliminate these route, whether actually it is working, so the residence time of the ophthalmic product we can determine with the help of this technique. And even if you are preparing the nasal product, again here the mucociliary clearance is again the clearance mechanism. Whatever the strategy you are using, so whether strategy is workable, that thing we can determine with the help of the gamma scintigraphy. Although there are some in vitro techniques are available, but again this is the additional data or even the actual images of the product performance after its application. So overall, these all images are indicating how the gamma scintigraphy after the radio labeling, how the radiopharmaceutical product can be useful in the product development. These are the evidence. So I think if we are incorporating these techniques in the product development, it could be useful, and you are now strengthened with the data. You can tell strongly this is the product performance. None of the person cannot be denied about, because this is actual images. None of the person can deny your products or your strategy or your whatever the approach you are using for the enhancement of the drug viability is not workable. So this is all about. Thank you very much. Thank you, Dr. Mehan, for giving the opportunity for this wonderful session and sharing my view and point on this specific technique, gamma scintigraphy. And this technique is will be very useful for the all students and even for all the pharmaceutical company which are regularly working in the or they are which are regular working for the improvement of the viability of their pharmaceutical product. Thank you very much.
Thank you, Dr. Nitin.
Thank you, sir.
Thank you, Dr. Nitin, for this wonderful talk. And the listeners, if you have any query, please write in the inbox of YouTube chat and also visit our website www.isfcp.org. We again thanks to all listeners and Professor Dr. Nitin Sharma. Thank you, sir. Thank you so much.
Thank you very much, sir.